Hi, welcome to Tej Academy. My name is Prasanna Kumar Reddy. Today, in this video, I am going to discuss an important area for all 12th PUC biology students. ಸೊ ನೀವೆಲ್ಲ ಪ್ರಾಕ್ಟಿಕಲ್ ಎಕ್ಸಾಮಿನೇಷನ್ ಬಗ್ಗೆ ತುಂಬ ವರಿ ಆಗ್ತಾ ಇದ್ದೀರಾ. ಐ ಹ್ಯಾವ್ ಸೀನ್ ಲಾಟ್ ಆಫ್ ವಿಡಿಯೋಸ್, comments given by different kind of students. Many videos are already available in YouTube to score out of 30 out of 30. So ಆ ಥರ 30 out of 30 ನಮ್ಮ ವಿಡಿಯೋ ನೋಡಿದರೆ ಓನ್ಲಿ ಬರಲ್ಲ. ದಟ್ ಈಸ್ ನಾಟ್ ಶೋರ್. ಐ ಕೆನಾಟ್ ಗಿವ್ ಪ್ರಾಮಿಸ್. ಈಗ ನೀವು ಜನರಲ್ ಆಗಿ ಕಾಲೇಜ್ ಟೈಮಲ್ಲಿ ಲ್ಯಾಬ್ ಸರಿಯಾಗಿ ಮಾಡಿಲ್ಲ, ಲ್ಯಾಬ್ ಕ್ಲಾಸಸ್ಗೆ ಹೋಗಲಿಲ್ಲ, ಆರ್ ಯು ಕುಡ್ ನಾಟ್ ಹ್ಯಾವ್ ಫೆಸಿಲಿಟಿ ಟು ಡೂ ದ ಲ್ಯಾಬ್, ಬಟ್ ಯು ವಾಂಟ್ ಟು ಗೆಟ್ ಮ್ಯಾಕ್ಸಿಮಮ್ ಸ್ಕೋರ್ ಇನ್ ದ ಲ್ಯಾಬ್. ಔಟ್ ಆಫ್ 30 ಯು ಶುಡ್ ಸ್ಕೋರ್ ಮ್ಯಾಕ್ಸಿಮಮ್. ಸೊ ಅಂದರೆ ನೀವು ಒಂದಿನೂ ಲ್ಯಾಬ್ ಹೋಗಲಿಲ್ಲ ಅಂದರೂ ಬೈ ವಾಚಿಂಗ್ ದಿಸ್ ವಿಡಿಯೋ ಯು ಕ್ಯಾನ್ ಕಂಫರ್ಟಬಲಿ ಗೆಟ್ 25+. ದ್ಯಾಟ್ ಮಚ್ ಐ ಕ್ಯಾನ್ ಅಶ್ಯೂರ್. ಸೊ ಓಕೆ, ವಿತೌಟ್ ಟೇಕಿಂಗ್ ಮಚ್ ಟೈಮ್, ಲೆಟ್ ಅಸ್ ಮೂವ್ ಟು ದಿ ವಿಡಿಯೋ. ಈ ವಿಡಿಯೋದಲ್ಲಿ ನಾವು ಫಸ್ಟ್ ಲ್ಯಾಬ್ ಎಕ್ಸಾಮ್ ಪ್ರಾಕ್ಟಿಕಲ್ ಎಕ್ಸಾಮ್ ಪ್ಯಾಟರ್ನ್ ಏನಿದೆ, ಎಷ್ಟು ಕ್ವಶನ್ಸ್ ನಿಮಗೆ ಕೊಡ್ಬೋದು, ಆ್ಯಂಡ್ ಹೌ ಯು ಕ್ಯಾನ್ ಆನ್ಸರ್ ದೆಮ್, ವಾಟ್ ವಿಲ್ ಬಿ ದ ಎವಾಲ್ಯೂಯೇಷನ್ ಸ್ಕೀಮ್, ಅದನ್ನ ಡಿಸ್ಕಸ್ ಮಾಡಿ ದೆನ್ ಐ ವಿಲ್ ಆಲ್ಸೋ ಗಿವ್ ಯು ಆಲ್ ಪಾಸಿಬಲ್ ಕ್ವಶನ್ಸ್ ಫಾರ್ ದಿಸ್ ಲ್ಯಾಬ್ ಕ್ವಶನ್ಸ್. ಸೊ ನಿಮ್ಮ ಲ್ಯಾಬ್ ಎಕ್ಸಾಮಲ್ಲಿ, ಬಯಾಲಜಿ 12th ಕ್ಲಾಸ್ ಲ್ಯಾಬ್ ಎಕ್ಸಾಮಲ್ಲಿ ದೇರ್ ಆರ್ ಟೋಟಲ್ 8 ಸೆಕ್ಷನ್ಸ್. ಈ 8 ಸೆಕ್ಷನ್ಸಲ್ಲಿ ಯು ಸಿ ದ 7th one ಆ್ಯಂಡ್ 8th one. 7th one ಅನ್ನೋದು ಥಿಯರಿ ಆ್ಯಂಡ್ ಪ್ರಾಕ್ಟಿಕಲ್ ಕ್ಲಾಸ್ ಅಟೆಂಡೆನ್ಸ್. ಎನಿವೇ ದಿಸ್ ವಿಲ್ ಬಿ ಗಿವೆನ್ ಬೈ ಯುವರ್ ಕಾಲೇಜ್, ಸೊ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಗೆಟ್ 5 marks. If you attend your lab exam, almost you will get 5 marks. The second one, completed and duly certified practical record. ಇದನ್ನು ನೀವು ಸಬ್ಮಿಟ್ ಮಾಡಿದರೆ, ಲ್ಯಾಬ್ ರೆಕಾರ್ಡ್ ಸಬ್ಮಿಟ್ ಮಾಡಿದರೆ, ಆಲ್ಮೋಸ್ಟ್ ಇಯರ್ ಆಲ್ಸೋ ಯು ವಿಲ್ ಗೆಟ್ 5 marks. So majority of the cases student will get out here 100%, maximum cases with few exceptions. So ಈಗ ನೀವು practical exam day you have to focus on this first 6 questions only. ಈ first 6 questions ಅಲ್ಲಿ the first practical is preparation of a temporary slide. This is something different for every student. ಇದು ಒಂದು ಡಿಫ್ರೆಂಟ್ ಪ್ರಾಕ್ಟಿಕಲ್ ಇರುತ್ತೆ. ದ ಸೆಕೆಂಡ್ ಒನ್, ಪ್ರಿಪರೇಷನ್ ಆಫ್ ಟೆಂಪರರಿ ಸ್ಲೈಡ್ ಆಫ್ ಮೈಟೋಸಿಸ್, ವಿಚ್ ಈಸ್ ಎ ಕಾಮನ್ ಪ್ರಾಕ್ಟಿಕಲ್ ಫಾರ್ ಥ್ರೂಔಟ್ ಕರ್ನಾಟಕ. PUC ಬಯಾಲಜಿ ಪ್ರಾಕ್ಟಿಕಲ್ ಅಟೆಂಡ್ ಆಗ್ತಾ ಇರೋ ಸ್ಟೂಡೆಂಟ್ಸ್ ಎಲ್ಲರಿಗೂ ದಿಸ್ ಈಸ್ ದ ಕಾಮನ್ ಪ್ರಾಕ್ಟಿಕಲ್. ಅಂದರೆ 5 marks. 5 marks ಆರ್ ಕಾಮನ್ ಫಾರ್ ಆಲ್ ಆಫ್ ಯು. ಇದು ಡಿಫರೆನ್ಸ್ ಇರಲ್ಲ. ದೆನ್ ದ ಥರ್ಡ್ ಒನ್, ಐಡೆಂಟಿಫಿಕೇಶನ್ ಆಫ್ ಪೆಡಿಗ್ರಿ ಚಾರ್ಟ್. ಸೊ ಒಂದು ಪೆಡಿಗ್ರಿ ಚಾರ್ಟ್ನ ನಿಮ್ಗೆ ಕೊಡ್ತಾರೆ, ಯು ಹ್ಯಾವ್ ಟು ಐಡೆಂಟಿಫೈ ಆ್ಯಂಡ್ ಕಮೆಂಟ್. ದಿಸ್ ಈಸ್ ಆಲ್ಸೋ ಗೋಯಿಂಗ್ ಟು ಬಿ ಕಾಮನ್ ಕ್ವಶನ್, ಬಟ್ ದೇರ್ ಆರ್ 5 ಡಿಫರೆಂಟ್ ಪೆಡಿಗ್ರಿ ಚಾರ್ಟ್ಸ್ ಪಾಸಿಬಲ್. ಅದು ಒಂದು ಸೆಂಟರಿಂದ ಒಂದು ಸೆಂಟರ್ಗೆ, ಒಂದು ಬ್ಯಾಚಿಂದ ಒಂದು ಬ್ಯಾಚ್ಗೆ ಡಿಫರೆನ್ಸ್ ಇರ್ಬೋದು. ಐ ವಿಲ್ ಡಿಸ್ಕಸ್ ಅಬೌಟ್ ಆಲ್ 5 pedigree charts so that you can easily score 3 marks. Identification of D and comment. ಇದರಲ್ಲಿ ಏನು ಪಾಸಿಬಿಲಿಟೀಸ್ ಇದಾವೋ, ಹೌ ಟು ಗೆಟ್ ದಟ್ 3 marks we will discuss now. So total 5 plus 3 plus 3, 8 marks. Identification of specimen E, ಇದರಲ್ಲಿನೂ ಏನು ಪಾಸಿಬಿಲಿಟೀಸ್ ಇದಾವೋ ಅದನ್ನು ನಾವು ಡಿಸ್ಕಸ್ ಮಾಡೋಣ. ಆ್ಯಂಡ್ ಐಡೆಂಟಿಫಿಕೇಶನ್ ಆಫ್ F ಆಲ್ಸೋ ವಿ ವಿಲ್ ಡಿಸ್ಕಸ್. ಓವರಾಲ್ ಈ ವಿಡಿಯೋದಿಂದ ನಿಮಗೆ ಆಲ್ಮೋಸ್ಟ್ ಫ್ರಮ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ 2 ಇಂದ, ಫ್ರಮ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ 2 ಇಂದ 6ವರೆಗೂ ಏನೇನಿದಾವೋ ಅದನ್ನು ವಿ ವಿಲ್ ಟ್ರೈ ಟು ಕವರ್ ನಾವು. ಸೊ ಲೆಟ್ ಅಸ್ ಮೂವ್ ಟು ದಿ ನೆಕ್ಸ್ಟ್ ಸ್ಲೈಡ್. ಸೊ ದಿಸ್ ಈಸ್ ಯುವರ್ ಫಸ್ಟ್ ಕ್ವಶನ್ ಇನ್ ದಿ ಪ್ರಾಕ್ಟಿಕಲ್. ಸೊ ಇಲ್ಲಿ ಫಸ್ಟ್ ಕ್ವಶನಲ್ಲಿ ಏನು ಕೊಟ್ಟಿದ್ದಾರೆ, ಪ್ರಿಪೇರ್ ಎ ಟೆಂಪರರಿ ಸ್ಲೈಡ್ ಟು ಶೋ ಪೋಲನ್ ಜರ್ಮಿನೇಷನ್ ಫ್ರಮ್ ದ ಗಿವನ್ ಮೆಟೀರಿಯಲ್ ಎ ಆ್ಯಂಡ್ ಕ್ಯಾಲ್ಕುಲೇಟ್ ದಿ ಪರ್ಸಂಟೇಜ್ ಆಫ್ ಪೋಲನ್ ಜರ್ಮಿನೇಷನ್. ಸೊ ಇದು ಒಂದು ಪ್ರಾಕ್ಟಿಕಲ್ ಇರ್ಬೋದು. ಆರ್ ದ ಸೆಕೆಂಡ್ ಒನ್ pollen tube growth practical, or the third one you need to prepare a temporary slide of given material A by taking TS of ovary, or the fourth one temporary slide to show nucleus staining from the given material. So idu in your examination center they may keep it as something like A1, A2, A3 or A4 materials. They may give you on lottery basis. So one student may get one practical. So illiro 4 practical na every student must read the process at least. ಈಗ ನಿಮಗೆ ಪ್ರಾಕ್ಟಿಕಲ್ ಮಾಡೋ ಫೆಸಿಲಿಟಿ ಇಲ್ಲ, ನಾವು ಯು ಕೆನಾಟ್ ಡೂ ಎನಿಥಿಂಗ್. ದಟ್ ಈಸ್ ವೈ at least for writing part, what is the aim of the practical process, materials required, etc. you should learn for these 4 practicals so that you will get 2 marks minimum. At least for preparation process, already lot of people made videos on this. You just choose anyone and go through it. Minimum nimge ee 3 marks alli 2 marks bartave. So out of 5 we can consider 4 marks scoring is easy. Coming to the second practical, prepare a temporary slide of the given material B and identify any one stage of mitosis. This is the common practical for throughout state. ಇದರಲ್ಲಿ ಮಾರ್ಕಿಂಗ್ ಸ್ಕೀಮ್ ಹೆಂಗಿದೆ, ಫಾರ್ ಪ್ರಿಪರೇಷನ್ಗೆ 3 marks and ರೈಟಿಂಗ್ ಕಾಮೆಂಟ್ಗೆ 2 marks. ಸೊ ಲೆಟ್ ಅಸ್ ಇಮ್ಯಾಜಿನ್ ನೀವು ಈ ಪ್ರಾಕ್ಟಿಕಲ್ನ ಕಾಲೇಜಲ್ಲಿ ಪರ್ಫಾರ್ಮ್ ಮಾಡಿಲ್ಲ, ಸ್ಟಿಲ್ ಯು ಕ್ಯಾನ್ ಗೆಟ್ 2 marks ಬೈ ರೈಟಿಂಗ್ ದ ಕಮೆಂಟ್. ನೀವು ಮಾಡಿರೋ ಸ್ಲೈಡಲ್ಲಿ ಮೆಟಾಫೇಸ್ನ ತೋರಿಸ್ಬೇಕು. ಸೊ ಹೌ ಡು ಯು ರೈಟ್ ಅಂಡ್ ಐಡೆಂಟಿಫೈ ದಿಸ್ ಆಸ್ ಎ ಮೆಟಾಫೇಸ್? ಸೊ ಇಫ್ ಯು ಐಡೆಂಟಿಫೈ ದಟ್ ಸ್ಟೇಜ್ ಆಸ್ ಮೆಟಾಫೇಸ್, ಸಿಂಪಲ್ ಆಗಿ ಯು ಟ್ರೈ ಟು ರೈಟ್ ದಿಸ್ 3 points. ನ್ಯೂಕ್ಲಿಯರ್ ಮೆಮ್ಬ್ರೇನ್ ಈಸ್ ಡಿಸಪಿಯರ್ಡ್. ಆಲ್ರೆಡಿ ಮೆಟಾಫೇಸಲ್ಲಿ ನ್ಯೂಕ್ಲಿಯರ್ ಮೆಮ್ಬ್ರೇನ್ ಡಿಸಪಿಯರ್ ಆಗಿರುತ್ತೆ. ಆ್ಯಂಡ್ ಈಚ್ ಕ್ರೋಮೋಸೋಮ್ ಈಸ್ ಥಿಕ್, ಕ್ಲಿಯರ್ಲಿ ವಿಸಿಬಲ್ ವಿತ್ 2 chromatids. ಸೊ ಒಂದು ಕ್ರೋಮೋಸೋಮ್ಗೆ 2 chromatids ಇರ್ತವೆ. ಬೋತ್ chromatids ಆರ್ ಜಾಯಿಂಡ್ ಎಟ್ ಸೆಂಟ್ರೋಮಿಯರ್. ಸೊ ಆ ಥರ ವಿಸಿಬಿಲಿಟಿ ಇರುತ್ತೆ. ಆ್ಯಂಡ್ ದೇ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಫಾರ್ಮ್ ಎ ಮೆಟಾಫೇಸ್ ಪ್ಲೇಟ್. Formation of metaphase plate. So in a cell, all chromosomes are present at center. So itara all chromosomes are present at center by forming a metaphase plate or equatorial plate. So this is very, very important point to identify this as a metaphase. So ಈ ಥರ 3 pointsನ ಇಫ್ ಯು ರೈಟ್ ಯು ವಿಲ್ ಗೆಟ್ 2 marks. ಸೊ ಪ್ರಾಸೆಸ್ಗೆ 3 marks ಆ್ಯಂಡ್ ಕಮೆಂಟ್ಗೆ 2 marks. ಸೊ ನಿಮಗೆ 2 marksಗೆ ಏನು ಬೇಕೋ ಅದನ್ನು ನಾನು ಇಲ್ಲಿ ಡಿಸ್ಕ್ರೈಬ್ ಮಾಡಿದ್ದೇನೆ. ಇಫ್ ಯು ಐಡೆಂಟಿಫೈ ದಿಸ್ ಆ್ಯಸ್ ಅನಾಫೇಸಲ್ಲಿ ಸಪರೇಷನ್ ಆಫ್ ಕ್ರೊಮ್ಯಾಟಿಡ್ಸ್ is the major event. Because of that, daughter chromosomes will appear in different shapes. V shape irbodu, J shape irbodu, I shape irbodu or L shape irbodu, depending on the location of the centromere, position of the centromere. Ee eradu points, these 2 points are the identification points for anaphase. Similarly for telophase, daughter chromosomes will reach to the opposite poles. So one cell alli already daughter chromosomes opposite poles reach ಆಗಿರ್ತವೆ. Nuclear membrane reappears and you can clearly see 2 daughter nuclei. So ಒಂದೇ cell ಅಲ್ಲಿ 2 nucleus ನೋಡ್ಬೋದು. That is the confirmation point for telophase. So ನಿಮಗಿರೋ ಮೈಟೋಸಿಸ್ ಸ್ಟೇಜಸ್ ಅಲ್ಲಿ prophase, metaphase, telophase, anaphase. So prophase, other than prophase, you can identify any 3 phases, that is metaphase, anaphase or telophase. Comfortably you can get that 5 marks. Now the most important thing, the third question. ಇದು ಮಾಡೋಕ್ಕೆ ನಿಮಗೆ ಪ್ರಾಕ್ಟಿಕಲ್ ಮಾಡೋ ನೆಸೆಸಿಟಿ ಇಲ್ಲ. ಇಫ್ ಯು ನೋ ದ ಕಂಟೆಂಟ್ ಯು ಕ್ಯಾನ್ ಗೆಟ್ ದಿಸ್ 3 marks. What is the third question? identify and comment on the given pedigree chart. So ಒಂದು pedigree chartನ ಪ್ಲೇಸ್ ಮಾಡಿರ್ತಾರೆ. that pedigree chart need to be identified. So identificationಗೆ 1 mark, correct identification, and commentಗೆ 2 marks. So 1 mark for identification and commentಗೆ 2 marks. So I will help you to get out in this section. So let us imagine this is one pedigree chart given in your lab manual. I need to identify this, what kind of pedigree it is, and related to this some comments. So let us see if I want to identify this as, first of all I should decide whether it is a dominant trait or recessive trait. Here in the pedigree I can clearly see parents are affected and they are producing affected children. Affected means disease. In a simple word, if parents are affected parents are producing affected children, that means it is a dominant trait. We can simply consider like this. So this is a dominant trait. So I have to confirm whether it is a autosomal dominant or X-linked dominant. Only 2 possibilities are there. So first point ನಾನು ಯಾವ ಬೇಸಿಸ್ ಮೇಲೆ ಇದು ಡಾಮಿನೆಂಟ್ ರೈಟ್ ಅಂತ ಕನ್ಫರ್ಮ್ ಮಾಡಿದೀನಿ? It is because of affected parents produced affected offsprings. This is the point I considered. Now there are 2 possibilities, autosomal dominant or X-linked dominant. If it is X-linked dominant, if it is an X-linked dominant, you can see here, for example, if it is a X-linked dominant, this father genotype will be XY. If this disease is because of the X chromosome, he cannot produce a affected son. If this disease is X-linked dominant, son cannot be produced by affected father. It requires mother, because this fellow will get X chromosome from the mother, not from the father. He will donate only Y chromosome. So from this area we can confirm this as autosomal dominant. So I am going to fill this as autosomal dominant. Once again I am explaining. ನಾನು ಫಸ್ಟ್ ಕ್ರೈಟೀರಿಯಾ ಏನು ತಗೊಂಡಿದ್ದೀನಿ, ಪೇರೆಂಟ್ಸ್ ಅಫೆಕ್ಟೆಡ್ ಆಗಿದ್ದಾರೆ, ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ಅಫೆಕ್ಟೆಡ್ ಆಗಿದ್ದಾರೆ. ದಟ್ಸ್ ವೈ ಐ ಆಮ್ ಥಿಂಕಿಂಗ್ ದಿಸ್ ಆಸ್ ಡಾಮಿನೆಂಟ್ ರೈಟ್. ಸೊ ವೆನ್ ಐ ಕನ್ಸಿಡರ್ ದಿಸ್ ಆಸ್ ಡಾಮಿನೆಂಟ್ ರೈಟ್, ಐ ಹ್ಯಾವ್ 2 ಆಪ್ಷನ್ಸ್. ಒನ್ ಈಸ್ ಆಟೋಸೋಮಲ್ ಆರ್ ಎಕ್ಸ್ ಲಿಂಕ್ಡ್. Autosomal, yes, mother is affected, producing son as well as daughter equally. Possibility is there, but it is also possible that she can be X-linked dominant also, because mother donates X chromosome for both. So I cannot simply conclude this. Next, secondly, illinu mother producing affected son and daughter. This is also possible, just like the first one. But if I come to the third generation, in third generation, affected person is only father. Affected person is only father, but he also produced affected daughter as well as son. So affected daughter father ಇಂದ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರ್ಬೋದು, but affected son, if it is X-linked, it is not possible. So this confirms that it is a autosomal. ಸೊ ಇದಕ್ಕೆ ನಾನು ಇನ್ನೊಂದು 2 ಪಾಯಿಂಟ್ಸ್ ಆ್ಯಡ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಏನು ಬರೀಬೋದು? ಪೆಡಿಗ್ರಿ ಈಸ್ ವರ್ಟಿಕಲ್. ವರ್ಟಿಕಲ್ ಅಂದ್ರೇನು? ದೇರ್ ಈಸ್ ನೋ ಸ್ಕಿಪ್ಪಿಂಗ್ ಆಫ್ ಜನರೇಷನ್. ಇನ್ ಎವ್ರಿ ಜನರೇಷನ್ somebody is deceased. That is why it is called as vertical. No skipping of generation. How I am confirmed this? Affected father produced both affected daughter and affected son. That is why I am confirming this as autosomal dominant trait. Of course, it is present in both the genders. Example. So one example we can consider for this. Polydactyly, having more than 5 fingers. A dimple cheek. So dimple cheek is also a autosomal dominant trait. For example, if this kind of pedigree is given, ಈ ಥರ pedigree ಕೊಟ್ಟಿದ್ರೆ, so ಈಗ parents are normal but producing affected offspring. So from this I can say this is a recessive trait. So when I say it is recessive trait, it can be autosomal recessive or X-linked recessive. Two possibilities are there. Now I have to confirm what type it is. For example, if it is X-linked recessive, let us imagine X-linked recessive. This mother is X-linked recessive, small c is there. She can produce an affected son, but she cannot produce an affected daughter because here father is normal. Is in the dot symbol carrier is showing, but for X character he is normal. So from this we can confirm that it is autosomal recessive. So we know that if it is X-linked recessive, affected daughters are produced by minimum deceased fathers. To produce affected daughter, minimum we should have deceased father. It is not available, that's why it is a autosomal recessive. So you can see clearly both the genders are equally affected, that's why we can consider it as autosomal. First point ಏನು ಬರೀಬಹುದು ಇದಕ್ಕೆ? Normal parents produce affected offsprings, or you can also consider it as affected offsprings are produced from unaffected parents. That is the first criteria to identify recessive trait. Second criteria ಏನili? Affected mother produced both affected son and daughter. Generally, for X-linked recessive, to produce an affected daughter, minimum father must be deceased, mother must be carrier. But here it was not like that. So this is the confirmation point that I have taken it as autosomal recessive. Examples ಏನಮ್ಮ? Albinism, SCA, sickle cell anemia, thalassemia. Majority of the concepts which are given in your syllabus are related to this autosomal recessive trait. Let us move to the third one. Third one ಏನಿದೆ? Parents affected ಆಗಿದ್ದಾರೆ and producing affected offspring. Okay. So that means it is a dominant trait. So we can say that it is dominant trait. So there is no skipping of generations. Now whether it is an autosome or X-linked, we should confirm. Affected mother producing daughter as well as son. Affected mother producing son, daughter, son, daughter. Affected mother producing son, daughter. Affected father producing only daughters. You see here, in every case, mother is producing daughter as well as son. Mother is producing son as well as daughter, affected people. But if you take here, male is producing only affected females. What it indicates? This fellow is X-linked dominant gene is having. Always father donates X chromosomes to daughters only. That is why in this family only daughters are affected. Boys are not affected. Opposite to the first one. So this is a X-linked dominant trait. Careful ಆಗಿ ನೋಡಿ ಇಲ್ಲಿ affected father is producing 100% affected daughters. That is the confirmation for X-linked dominant. So first point is just like the previous one, affected parents produce affected offsprings. This is one, that's why we confirmed it as dominant. Second one, affected father produce 100% affected daughters, all affected daughters. This is the confirmation point for X-linked dominant trait. Example, DMD, Dutch name muscular dystrophy. This is the example for X-linked dominant trait. Let us see the fourth pedigree chart. So in this pedigree chart, we can clearly understand parents are normal but offsprings are affected. So from this, we can analyze this as recessive trait. So now we have to decide whether it is a autosomal recessive or X-linked recessive. So let us see, if it is autosomal recessive, usually it should present in both the genders, that is male as well as female, almost in equal distribution level. But if you see in this pedigree, majority of the cases males are affected. That is one point. The second point, everywhere normal parent, so father is normal, mother is a carrier producing a affected son. So carrier mother producing a affected son. Here also carrier mother producing a affected son. Here also carrier mother producing a affected son. Carrier mother producing a affected son. So this shows that it is a X-linked. One more third point, it is going to confirm, to produce a affected daughter, father must be affected. So this also confirming that it is a X-linked recessive trait. So we will confirm this as X-linked recessive trait. First point ಏನು ಬರೀಬೋದು, affected parents produce, sorry, not affected parents, normal parents. Normal parents produce affected offsprings. This is the common point when we consider something as recessive. The second common point is always normal parents are producing affected boy. So here a carrier mother and normal father. So normal parents are producing affected son basically. Affected daughter is produced when the father is affected. That will be the third point. So example for this will be hemophilia, color blindness. So both we can consider it as X-linked recessive. So let us move to the next one, the last pedigree chart, fifth one. Here you can see in this pedigree chart, only males are affected. So from father to son, again father to son. So since it is transmitting from male to male, so we can consider this as Y-linked trait. So Y-linked trait inheritance observed in only males. So this is observed only in males. And the second point, affected father produces 100% affected sons. So affected father produce 100% affected sons, not daughters. All affected sons. So that will be the confirmation point for this. So example of this is hypertrichosis, growth of hair on ear pinna. Hypertrichosis is a Y-linked trait. So if you remember these 5 charts and their techniques, you will easily get 3 marks. Similarly, coming to the 4th question, identify D by giving 2 reasons. So D ano material na now identify ಮಾಡಬೇಕು and we should give 2 reasons for that. So in D material ge what are the possibilities? Maybe you may get a TS of mammalian testis or transverse section of mammalian ovary or VS vertical section of angiosperm ovule. You will get basically ovule, maybe anatropous, orthotropous, whatever it is, either in the form of a slide or in the form of a picture. So you should identify the given material and write at least 2 reasons why it is like that, why you are identifying that as the same material. For this you will get 3 marks. It doesn't require any practical experience, just you should understand the content and able to identify the pictures. Coming to the 5th question, identify the given specimen. Here they are going to give a specimen E and name the disease it causes. So 100% this specimen is a pathogen, basically disease causing agents. So 1 mark for correct identification, 1 mark for the name of the disease. So in your lab manual there are 4 different type of specimens which are related to disease causing. So one is Entamoeba histolytica. Entamoeba histolytica causes amoebic dysentery or amoebiasis. Plasmodium vivax causes malaria. Trichophyton rubrum causes ringworm. Ascarias lumbricoides causes ascariasis. So organism name and the disease caused. If you write both of these, you will get 2 marks. This will be the 5th one. So this usually they give pictures. This is the Entamoeba histolytica. And this is the Plasmodium vivax. This is the Trichophyton. Ascariasis they can give even the specimen, spottle. The final question, 6th one, identify F and comment on it. This is also for 2 marks. Identification 1 mark and commenting 1 mark. So ಇದರಲ್ಲಿ ಏನು possibilities ಇದೆ? For this F question, they can plan something, a picture of bagging, hybridization process, bagging, crossing, okay? Homologous organs in the plants or animals, analogous organs in the plants and animals. Any one you may get. So if you know the information of bagging, homologous organs and analogous organs, you can easily get 2 marks here. I will show you some simple example pictures, sample pictures. So if they show this picture, this is showing a cover, a bag, a polythene cover is attached to the flower. So covering the flower. So this is a bagging process. We know that bagging is meant for to control unwanted pollination basically. Next picture, you can see onion and opuntia. So onion stores food material in the leaves, scale leaves. Opuntia, leaves are converted into spines. So in both the cases, origin is same, leaf, but it is performing different functions. So when origin is same, performing different functions, they are called as homologous organs. So these are homologous organs in plants. Similarly, a human limbs as well as bat limbs, origin is same, their bone structure is same, but the function is different. In bat it is useful for flying, in human it is useful for walking and other locomotor structures. So origin is same, functions are different. So that is an example of homologous organs. Let us see the other one, ginger and carrot. Ginger, carrot. Ginger is a modified underground stem, whereas carrot is a modified root. So both are different structures. Origin is different, but their function is storing food material. So origin is different, function is same. So it is a analogous organs. We can name it as analogous organs in the plants. Similarly, a wing of bird, wing of insect. Wing of bird will have all bone structures similar to the vertebrates. But here, wing of insect do not have any bone structure. But in both the cases, wings are performing same function, fly. So function is same, origin is different. So this is also an example of analogous organs. So by this, I would like to say, even though you did not perform good practicals in your college, you never entered into laboratory, it doesn't matter. You can easily get minimum 25 plus marks if you are good at content and identification of the given pictures and specimens. It's quite easy. So I hope this video is useful for you. I feel that it is useful for you. If you like the video, please share with your friends who are appearing for the second PUC biology practicals. And to get more videos instantly when we release, please do follow our YouTube channel. For more queries or any career guidance, you can also contact us through this number. Thank you.